Hi, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce our next talk, Clinical Development of CRISPR-Directed Gene Editing for Solid Tumors. I'm pleased to welcome Kmiec as your presenter. Eric is the founder and executive director of ChristianaCare's Gene Editing Institute. He is widely recognized for his pioneering work in the fields of molecular medicine and gene editing. Throughout his professional career, Dr. Kmiec has led research teams in developing gene editing technologies and genetic therapies for inherited disorders such as sickle cell disease. In the basic research arena, his work helped elucidate the regulatory circuitry that controls gene editing of human cells. Current clinical research and development centers on developing CRISPR-based gene editing approaches for solid tumors, squamous cell carcinoma of the lung, and esophageal cancer, and the lead protocols currently advancing through the FDA approval process. A warm welcome to you, Eric. Following the presentation, we'll have a Q&A session, and we'll welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Eric's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon on the menu on the left-hand side of the player. Now, without further ado, I will hand over to Eric to begin his presentation.
Well, Carolyn, thank you very much, and good morning, good afternoon, good evening, everyone, wherever you're listening in. And I want to thank Technology Networks for this wonderful opportunity to talk to you today about some of the work we've been doing, and more importantly, the way we're beginning to think about using gene editing for targets in oncology. So I want to start first with where we are working. We work within a large health system located in northern Delaware that serves New Jersey, Maryland, southern Pennsylvania as well. And as many health systems are beginning to evolve, ChristianaCare is starting to push further and further into research and innovation. And so while we have an outstanding primary care facility, as all hospitals do, we're now starting to move more into the next generation of healthcare, and that includes developing unique approaches to a variety of genetic diseases and in cancer. So I've been in the field of gene editing for quite some time. This was a cover article of the Journal of Clinical Investigation published in 1999, and I hasten to say that the people listed on this cover were approximately the number of people who were in the field at that time. I think we all know this has grown dramatically with a lot of outstanding folks joining, and we couldn't be happier that the field has now become a bit more mainstream in the scientific community. I formed the Gene Editing Institute in 2015 after having faculty positions at the University of California, Davis, Thomas Jefferson University, and the NCI Center of Excellence in Philadelphia, and then moving into the University of Delaware. In 2015, I moved my laboratory into the ChristianaCare Helen F. Graham Cancer Center, primarily because I felt it was time for us to start thinking about clinical applications of gene editing. And I felt there was nowhere better than to do that than at a community cancer care center where we see all kinds of patients of socioeconomic and ethnic backgrounds. We formed GEI basically with three fundamental missions. The first is discovery research, in which we try to continue to understand the molecular basis of gene editing, and that's funded by federal and non-federal grants. And we call that sort of our innovation zone. Importantly, I also recognize that if we're to deliver breakthrough technologies to minority communities, we're going to have to spend a great deal of time and energy in educating the next generation of scientists, but also to build trust in communities where trust has been abandoned in earlier iterations of introducing breakthrough medicines. And then finally, the one that we'll focus on today more is our translational research group. This scientific advance in the field of gene editing is now beginning to approach clinical application. And we've been in part of the team that's certainly been in the forefront of translating things from bench to bedside. And I'm going to tell you today a little bit about our successes and our challenges that still remain moving this into a lung cancer application. First, I just want to use some slides I've used for many years, kind of describing exactly what gene editing is and how we think about it. It's very complicated, as any molecular process is, but we think about it as a genetic spell check. So when you receive an email or a document, hopefully people have cut, pasted, or changed words that were improperly spelled. We can remove them and change them and rewrite them. Well, gene editing is fundamentally born out of the same concept. We can cut out a gene and we call that a deletion or a knockout. We can insert genes or paste genes in, or sections of genes, and we call it insertion. And we can replace genes and knock genes into certain sites within genes. Deleting a gene right now and cutting it out is almost standard of practice within the animal model system. Inserting genes is a little more challenging, particularly at the sites where we want them to go. And replacing genes still remains a bit of a dream right now, although good progress is being made. So knowing this, we decided to focus all of our attention clinically on using these genetic tools that I'll talk about in a second for deleting a gene or knocking out a gene. We focus almost exclusively on the CRISPR-Cas system. So CRISPR is a programmable nuclease system that originates out of bacteria and is the adaptive immune system of bacteria. So when a phage or another type of parasite invades a bacterial cell, amazingly, the bacterial cell has evolved a whole series of CRISPR pathways whose main function is to disable that gene, but not only disable it, but take bits and pieces of the phage genome and put it into the bacterial genome. So the next time that phage comes and infects the cell, that bacterial cell is ready to fight it off, akin to the way we do when we get vaccinated and when antibodies are in place in our body to fight off the subsequent viral infection. The CRISPR system has now taken over the gene editing world. There are many reasons for this, and many great folks have entered the field. But essentially, it's basically for two reasons in my mind. The first is it's naturally occurring. So evolution has given us a gene editing tool. And the second is it's made up of two just very simple components. A CRISPR guide RNA, which is represented by the little gold man, and a Cas nuclease, or molecular scissors that the gold man is carrying. The gold man walks along the DNA and moves the scissors to a specific site that we have predetermined and pre-designed into the gold man to make that cision. But keep in mind the CRISPR-Cas system does one thing. It cuts DNA. To repair DNA or insert or to replace genes, that's going to need other components. Perhaps that's why it's been a bit more challenging. But if we were to knock a gene out, whether it's in animal cells or now hopefully in clinical applications, we feel that the CRISPR-Cas system is the true genetic answer. So now I want to move a little bit more into an area that we thought about for quite some time. As I've mentioned, I've been in the field for 25 to 30 years. I've seen lots of different attempts and failures and hype surrounding gene editing. And I sort of know where all the potholes in the road to success are located. One of the most important things about developing a clinical application for any sort of genetic medicine is to think carefully about the target choice and the target gene. And that's what we began to do in 2015 when we entered the Helen F. Graham Cancer Center and began to go to grand rounds and surgical meetings and listen to oncologists talk about where their shortcomings were. So the entire program we've built here at GEI is based on organically, from the ground up, listening to clinicians first and then applying a technology. In many cases, technology is developed and then it goes in search of a target disease. We sort of did it in opposite ways. We had a good technology, but we wanted to build that technology into the decision process of how we choose the target disease. And so we started looking at lung cancer for a couple of reasons. First, lung cancer is a deadly disease. It's the #1 cancer killer in the country, the United States, and in the world, and certainly in the state of Delaware. This is no surprise. We've dealt with lung cancer for a long time, and it has some elements clearly that exacerbate its appearance and its deadly consequences. But it was also the point that very little is known about how to treat lung cancer effectively. While we have great hopes for immunotherapy and T-cell therapy and CAR-T therapy, we're now learning that immunotherapy, particularly using T-cells, is going to have a challenge for all solid tumors because of the matrix of the tumor prevents equal access of those T cells. And in some cases, the tumor has figured out small peptides to deactivate T cell activity. We have great hopes with that, and we're very good friends with folks at the University of Pennsylvania. We're rooting for them. But lung cancer has very few options. And so we started to think about how could we, using CRISPR, begin to affect the treatment of lung cancer, and how could we build a therapy to treat them? Lung cancer is divided into two major groups, small cell and non-small cell lung carcinoma. And the non-small cell lung carcinoma group are almost equally divided between adenocarcinoma and squamous cell carcinoma. So 31% versus 41%. That's a lot of people, no matter what percentage it is. This is a slide provided to me by Greg Masters, who's a surgical oncologist and the principal investigator of our NIH Center of Excellence grant in lung cancer. And we can kind of look at all of the great work that's going on. And in a pressing way, we see that some of the major targeted therapies like Keytruda or Opdivo are effectively appropriate only for about 30 to 40% of the patients. And in the case of Keytruda, that's probably drops down to 19 to 20%. And we've recently seen that Keytruda, which was initially a very strong drug for lung cancer alone, is now being coupled with chemotherapy or radiation in a combinatorial approach. And that caught our attention because while Keytruda and other drugs are very effective for short times, in the long term, it does appear as if a supplemental treatment, an augmentary therapy, might actually be producing the best results. And then we noticed on the right side of the screen that 25% of all lung cancer patients, no matter what they are, no matter what their source, are completely unavailable for targeted therapies. And that's because they lack the famous biomarkers that sit on the cells of the tumor cells. And at the bottom of this particular slide, in each of the three lines, you see the word chemotherapy. In many cases, despite our greatest attempts and what a lot of people want to have true, chemotherapy does still remain to be a significant part of the treatment of lung cancer. And obviously, when chemotherapy comes forward, chemo resistance is not far behind. So we chose lung squamous carcinoma after about a year of research and discussions and meeting with surgical oncologists. Because it's less studied, there are very few treatment options, and drugs like Keytruda and Opdivo failed in targeted therapy. There's poor response and prognosis, and a median OS is much shorter than its sister adenocarcinoma. So we thought here was a place that we could apply a unique targeted therapy approach to perhaps help in the overall treatment of lung cancer. So the second thing you need to think about when you construct a gene editing program for any sort of clinical application is your target gene. Now, many of my colleagues through the years have used CRISPR and TALENs and zinc finger nucleases to try to correct mutations that occur in tumor suppressor genes like p53. And while that has shown some promise in cell models and a little bit in animal models, it's an endless game of chasing your tail because p53 mutations occur as the tumor grows under stress and pressure, and the mutator phenotype actually begins. And so you're always trying to repair and correct a mutation, but you never really catch up. And remember in one of the earlier slides, I mentioned the gene repair or fixing a mutation, inserting or replacing a gene is actually far more difficult than knocking out. So bearing in mind squamous cell carcinoma of the lung and the fact that CRISPR is a naturally occurring knockout device, I chose to work on the NRF2 KEAP1 pathway. Now, ironically, KEAP1 is the target for Opdivo and Keytruda. And so KEAP1 has already been worked on well and modest success as we previously discussed. But NRF2 remains a bit of a mystery. There are very few targeted therapies for it, and there are very few drugs in the pipeline. It's not clear why that is, but one reason is that NRF2 is not truly amenable for the development of small peptides or targeted therapy. And then in 2008, a group at Arizona State University, led by Donna Wang, reported a fascinating observation. She noted that NRF2 has two sides, and sometimes known as the dark side of NRF2. In some cases, NRF2 allows you to withstand stress, whether it's being outside in the sunlight or an admission of some toxic chemical, smoking. NRF2 levels protect you essentially from responding badly to stressful conditions. That's all well and good until a physician, an oncologist, are trying to treat you with radiation or chemotherapy for a lung tumor. Now the NRF2 is our enemy because it now starts to protect against the effectiveness of the chemotherapy. And that caught our eye. So we questioned, is NRF2 a friend or an enemy? And a couple of very interesting papers about 15 to 20 years ago also indicated that NRF2 activity starts to move forward on cancer tissues as they enter later stage. I've talked a little bit about the multiple functions of NRF2 on the left-hand part of this slide, and the key, of course, for us is detoxification. But on the right-hand side is the expression levels, mRNA levels, of a target gene of NRF2. NRF2 is a master regulator, a transcription factor that controls about 100 to 200 genes. Some people believe 300. And one of these is the gene illustrated here. And what we noticed was as time went on in cancer tissues, from stage 1 to stage 2 to stage 3, and this is lung cancer, the amount of NRF2 activity increases as a function of the expression of downstream genes. And that told us that, first of all, NRF2 is regulating the response to cancer and to chemotherapy, but also seems to be elevated and protects the cell further. So as you get deeper into the stages of cancer, NRF2 is fighting off even more aggressive treatment. And it's that where we thought we could help. Other people had actually seen this a little bit before and had thought about how to knock down NRF2. And here's a series of very elegant experiments in which NRF2 was knocked down by siRNA. And you can see that as cisplatin, doxorubicin, and etoposide concentration curves increase, cell viability is decreased if the NRF2 is not there. And these are tumor cells. It started to regulate to us that maybe NRF2 would be a good target. And so we started to think about using CRISPR, not to just disable partially as in targeted therapy or siRNA therapy, but rather to disable the gene at the genetic level. So a large percentage of the cells would not be able to produce the one thing that's preventing standard of care therapy from working. So how did we go after that? So one of the things that CRISPR has been known to do very effectively, as we talked about, is that it is enabling a genetic knockout of a gene. And so we thought about what's the best way to show that we could effectively knock out NRF2 in lung cancer cells and establish a paradigm that we might be able to advance toward clinical application. So here are a series, a summary of experiments that were done in 2017, 18, and 19 that shows kind of the same reaction you saw with siRNA. On the left-hand side is a kill curve, increasing concentration of cisplatin with a gene that has been, with a cell line that has been knocked out of NRF2, that's 211, or a wild type lung cancer cell. And you can see if you disable NRF2, the sensitivity to chemotherapy is increased dramatically and chemo resistance reduces. On the right-hand side is a picture of the tumors. And this is a pretty standard picture we saw for years, where if you just have lung cancer cells growing in saline or treated with cisplatin, and again, there's the chemo resistance, there's basically no change. But if you start to knock the gene out, whether alone or in combination with cisplatin, going back to the augmentary or combinatorial therapy, we saw a pretty significant cessation of growth and eventually cell killing of these tumors. Many of you are probably leading laboratories where you have talented young students And I've had a whole series of these. I've graduated 21 PhD students who, ironically, as you know, go off and be your main competition in the field of gene editing. And I'm very proud of them. But I also had a student in my lab, Kelly Banis, who was very much baked into the NRF2 project. And Kelly is a great young geneticist, and she noticed something interesting. And she noticed that in wild-type NRF2 genes, and genes that are in the bronchial cells and in normal somatic cells, there's a site called R34G that exists at the beginning of exon 2 that is just a sequence that a CRISPR molecule would not cut at. But she found that in mutant NRF2, in lung cancer cells, a unique site is naturally created by the mutation rate or the mutator phenotype of the progression of oncogenesis. So these sites are created just in the normal mutation rate of a cancer cell. And we call this a PAM site. This is a motif that's absolutely required for CRISPR to cleave. And amazingly, a single base change, a C to a G in this case, enables a CRISPR which we can design to essentially cut the mutant NRF2 existing in lung cancer cells, but not cut the gene, the same gene at the same position in bronchial cells or in other kinds of somatic cells. This wanders into the zone of the specificity we've all been searching for in tumor cells. Can we create a drug that targets only a tumor cell or a cancer cell and not the normal cell? Here we're doing this at the genetic level, which is a unique approach for sure, but the activity of the CRISPR molecule is absolute. And so moving this forward, we should be able to develop a paradigm where perhaps we can design CRISPR to knock out the genomes of tumor cells and not normal cells. This is a slide that I've shown in many places that people still marvel at, and this is the work of Kelly Banis again. What she's showing here is a percent of indel formation as a result of CRISPR activity on normal cells and on tumor cells. Indel formation is a measure we use to detect the degree of activity of CRISPR. It's basically telling us this is a mutation rate that's caused by CRISPR. 0.015% of the normal cell genomes are cleaved by CRISPR. This is so low, we're just sort of estimating this because we almost never find a normal cell that is cut by this CRISPR molecule. A cell line that doesn't have this mutation is cut at 0.04%. But a lung cancer cell that has one mutated copy, a heterozygote at R34G, is cut 35.87%. And the home run was the homozygous R34G site where both or three alleles have this specific mutation. And we've demonstrated repeatedly that over 93% of those tumor genomes, those tumor cells, have an inactive NRF2. This has all been published. We have a couple more papers on the way. So we always put our data in the mainstream and really strong journals to make sure that we have getting a good scientific community response. So as we started thinking about the data that we've been seeing and sort of our results, is it possible that we could begin to develop a clinical approach to squamous cell carcinoma of the lung using a combinatorial therapy of gene editing to disable the NRF2 gene specifically in tumor cells and not normal cells, augmented by continual treatment of chemotherapy, which most of these patients are frankly on already. Now, squamous cell carcinoma at stage 2 has a primary location at the base of the trachea, and that was another factor in thinking about how we could go after solid tumors. We understand and appreciate that lung cancer is a quite metastatic disease, but if caught early and if we could disable the primary tumor, we could reduce the rate at which metastatic tumors arise. And even in later stage, if we can disable the primary tumor and surgically remove it, the patient would have a better quality of life. And I'll talk a little bit about our thinking about clinical endpoints later. But this was an idea that we started to think about and perhaps needed to do a little bit more work. So we started on a great number of mouse studies. And I'm not going to go through in great detail here, but just give you the take-home message. I know many of you have done these Kaplan-Meier survival curves and the work, and the number of mice involved in this are astronomical. But we were pretty encouraged because the probability of survival of all of our data indicate that if we do the combinatorial approach with R34G plus a low dose of chemotherapy, all of the mouse continue to survive for weeks and weeks and weeks. And we actually had to stop this experiment here because you can see when we don't use that combination, mice are beginning to die. And so our control groups essentially are dying off. And I was a little uncomfortable about continuing an activity curve when our control groups are disappearing. But suffice it to say that these data convinced us that it is possible to target R34G in squamous cell carcinoma, combine it with a low dose of chemotherapy by tail vein injection, and keep mice alive for a lot longer period if either of those two particular treatments were not there. We moved it forward to the FDA, and the FDA meeting was extremely exciting and positive, and we've had very informal conversations since then. And what the FDA recognized, this approach, this combinatorial approach of using genetic tools and standard of care therapy, whether it's chemotherapy, radiation therapy, perhaps even immunotherapy, might actually set a new type of approach for the treatment of solid tumors. So they asked us to expand our breadth and move into something known as a basket trial, where we have multiple indications of solid tumors. So as we move through the approval process, we have our eye specifically on lung squamous cell carcinoma of the lung, but we're also doing backup work in esophageal and certain kinds of head and neck cancers as well. We hope this sets a broad-based application or broad technology that will be sufficient to treat those kind of therapies, those kind of cancers in the same pattern. All right, so now I want to move to one of the big questions and something we've worked on very intensely over the last 6 months, and that is exactly what is the delivery mode and delivery vehicle. So systemic delivery of CRISPR by just injecting biomolecules into a patient has a great number of challenges, particularly if you use a viral vector. So we discarded that idea right away. And again, remember, we're trying to develop an innovative therapy for solid tumors. So we want to de-risk everything. We don't want to worry about how much gets to the tumor. We don't want to worry about immune responses. So we started to think about direct intratumoral injection, which is commonly done for solid tumors, specifically in the immunotherapy area. The NIH has a bunch of clinical trials ongoing in which a bronchoscope and a needle is used to inject biotherapeutic activities, even chemotherapy, radiation seeds into solid tumors. And we thought we're just going to piggyback on something that is already in practice and for which there is FDA approval. Interestingly, along this period of time, the FDA encouraged us to look other places, and believe it or not, they were right. NRF2 mutations, specifically R34G mutations, do appear to be in other forms of lung cancer. And so other targets are available even via this intratumoral injection. Although our focus remains on lung squamous carcinoma, we believe that this type of an approach via direct injection could actually be effective against other forms of solid tumors. But what do we package the CRISPR activity, the CRISPR complex in? About 2 years ago, a company called Intellia launched a very aggressive and innovative approach to treating a liver disease known as TTR. If you inject any non-viral particle into a human being systemically, in almost all cases, the vast majority of that non-viral particle, probably even viral particles, end up in the liver. And so they did the same thing we did. They mitigated their risk by asking, let's not worry about loading a specific particle into the bloodstream and hope that it goes to the brain or to the lung or to the heart. Let's take advantage of where it actually goes. And so they developed lipid nanoparticles that were directed to the liver to treat the TTR disease. And in fact, it's worked marvelously. New England Journal of Medicine articles are written about this, and they're actually already through second and third levels of clinical trials with low toxicity and no negative response. We jumped on it. We thought the lipid nanoparticle is actually the answer for delivering the CRISPR-Cas molecule. Now, many of you know that through the years, everyone, including myself, has hoped that some way we would be able to develop a non-viral delivery vehicle. And it's been impossible. Nanoparticles made of gold or silver or copper are toxic, and they do not release their payloads very well. But over the last 5 years, some pretty significant changes in the cationic ionizable lipid, the level of cholesterol, and importantly, a helper lipid were incorporated into these molecules. And now suddenly, we think we have an excellent opportunity to deliver these LNPs. And last, of course, the LNP was part of the COVID vaccines. So we've had the greatest clinical trial in the history of man, delivering mRNA with an LNP and with magnificent results, with certainly acknowledging some toxicity. But in our case, we thought this was absolutely the best delivery vehicle. So we formed a partnership with Precision Nanosystems, or PNI, located in Vancouver, Canada. And we asked them to send us a whole series of 10 specific LNPs that they have been using, some in clinical trials, some for transfecting tumor cells, et cetera. We were able to put mRNA and the CRISPR-RNA, the three components, together, and we started to test to see how these type of LNPs would be successful in delivering our specific molecule into the tumor. So we did a whole series of experiments with hundreds and hundreds of mice. And in the end, we felt that 3 LNPs, 3, 4, and 5, actually turn out to be keeping, by direct injection now, the fluorescently labeled LNP and mRNA in the tumor. High delivery, high levels of expression, which is very important. One of the questions they always get is the level of expression inside a tumor going to be sufficient? So far in the mice, it's very, very efficient, and we're very excited about it. So here was a series of indications that told us that maybe 3 or 4 or 5 of these might actually be useful as we move forward. Another important question is, where else does it go? So if you directly inject a tumor with an LNP containing a messenger RNA of the Cas protein and the CRISPR-RNA, our gene editing package, does it move anywhere else inside the mouse? And these are, of course, the biodistribution studies that are so important for clinical application and for FDA approval. Well, there's a lot of ways to do this, and I could show you a ton of different tissues, slices, and things, but figuring that this might not be the best venue to do that, I'm showing you a qRT-PCR analysis, which is an ultra-sensitive analysis of tissues we've collected 24 hours and now even 72 and 96 hours later after injection. And what we find is that the expression of these LNP molecules is very, very high within the tumor itself. Very exciting. We think this is absolutely enough. Notice the axis there, 50,000 as a unit for expression of a fluorescent marker. If we now move to the distribution throughout the body, tumor, lung, liver, ovary, brain, and spleen, especially ovary, the level of expression is essentially 0. Keep in mind, the x-axis is 5 instead of 50,000. So the level of expression here is almost non-detectable. And if we push it out to 96 hours and beyond, we see nothing. So right now, we're tentatively, cautiously optimistic that after injection of the LNP containing our CRISPR payload, the molecule is actually staying in the tumor. But what about efficacy? What about our famous indel formation? This is brand new data. We've just started a massive experiment, but here's some early data without optimization, without dosing or anything. And amazingly, we're starting to see 10 to 20 to 30% of gene knockout. This means that 30% of the tumor cells, and remember, not the normal cells that infiltrate or surround the tumor, bear the NRF2 knockout. We don't know what percent's really required, but there's an anecdotal story that gives me some hope that we might be getting close. In the early days of treating sickle cell disease, people used to tell us that we needed to correct a homozygous sickle patient to normal at 50% the level. And this was provided by great geneticists who had studied sickle cell disease for a long time. It turns out not to be true. The current work by other laboratories on sickle cell disease tells us that if we can convert fetal globin or correction to 15%, 15, not 50, the patient develops a carrier state, especially if they stay at sea level. So ultimately, the level of expression and the level of knockout is probably lower than we really imagine it's needed. But of course, our work will continue to press the envelope to get it up to a high level so that the chemotherapy will respond. Over the next 3 to 4 months, we will actually be conducting in an animal the actual clinical trial, combining gene editing with chemotherapy. So I wanted to kind of come down the home stretch now and talk to you a little bit about what's next. So the question becomes, okay, squamous cell carcinoma, a subset of squamous cell carcinoma that have that particular mutation, very similar to the immunotherapy response. We're adding another tool to the toolbox there. But it turns out that other kinds of cancers, melanoma, thyroid cancer, breast cancer, cervical, bladder, endometrial cancer, all have mutations in a certain percent of those cancers that could be attacked in the same way. Uniquely, that a CRISPR molecule targets the tumor genome of the cancer and not the normal cells that are surrounding it. And this is an important fact. There are normal cells surrounding the tumor. And so the distinction at the genetic level is even more important now, because a targeted therapy will very rarely distinguish between tumor and non-tumor cells, even if the protein is slightly changed. That's why all targeted therapies develop resistance. And that's why drug companies get the newest iteration of these drugs, including TK inhibitors, for example, every 2 to 3 years. Here, we're targeting the gene, disabling it in the tumor cell, so that problem is non-existent. As I've talked about, our strategy is to basically stay focused on a mitigated risk. So we're doing intratumoral injection of esophageal cancer and squamous carcinoma of the lung. But we do hope, with our friends at Intellia and other companies jumping into the area, that ultimately we will be able to do systemic delivery, perhaps with LNPs that target and locate at a high percentage to those specific tissues. Right now, that's not possible. So we have to be realistic again about what we can do with this. And unfortunately, there are plenty of patients who could be treated by our approach with intratumoral injection. I want to talk about one of the most important parts of gene editing, and that's what I call the genetic consequences of gene editing. I get a lot of questions about, okay, it's very, very specific for that site, and we believe you, and it doesn't target that gene in a normal cell, but what about other genes? Well, here's the problem. Even at the FDA, I would say that the work about understanding off-target mutagenesis is best described as we're building the plane as we fly it. We don't know the best way to access this information. Every 3 to 4 months, there's a new technology in sequencing and bioinformatics analysis. And so we continue to follow that. And so I think the answer is two-layered. One, choose a disease where off-target mutagenesis is not the biggest problem for the patient. In a child treated for sickle cell disease or cystic fibrosis, or even a pediatric form of cancer, if the CRISPR goes unnoticed and targets somewhere else, in 20 to 30 years, the genetic consequences of that for a child treated at age 3, let's say, could be consequential. But a patient that has stage 2 or stage 3 cancer, where the long-term objection is to give 6 to 8 months more of life, like the targeted therapies of Keytruda and Opdivo, off-target mutagenesis that would appear 20 to 30 years later, and in fact even forming another tumor, is probably not at their level. And the second way to think about it is that in some cases, CRISPR has been shown to have off-target sites, but there is no evidence that any of those off-target sites have ever caused any adverse effect in an animal model. Now, that doesn't mean that this is unpublished and we're going to see it next week or there's going to be some announcement. But right now, the detrimental effects predicted by off-target mutagenesis have not shown significant impact in the field. I've shown this slide before, and we continue to do this over and over again. Positive safety data so far. The CRISPR R34G targeting package we use in the LNP, and we've had to repeat all of this, of course, now because we've chosen the LNP, and our discovery and our validation phase continue to show no off-target mutagenesis, except for one small point that is not in a gene, it's in an region for which there is no transcript. We don't see any adverse effect, and clearly that's not accompanied. But of the 500 genes that have unique potential off-target sites, when we sequence them with the blue dots below, we see almost no activity of significance. So again, with our patient target base, and what we're trying to achieve here in augmentative therapy and killing cancer cells, this kind of level is actually quite acceptable, and it's probably acceptable for almost all kinds of therapies in the oncology space. And then finally, I want to show you a really nice piece of data that just came off the presses. We have a group of people that continue to ask questions about, okay, what does the knockout of NRF2 look like? So we've cloned out cells that have been targeted with CRISPR. And what we show here on the left-hand side is a very nice Western blot that tells us that in the homozygous C4425 cell line, in which NRF2 has been completely disabled, there are no functional copies. The protein kind of hangs around for about 72 hours and is lingering for 2 weeks, but it is completely non-functional because the sensitivity of chemotherapy has gone up. On the right-hand side, a very boring but important experiment. At 72 hours, 2 weeks, and 3 weeks after targeting, the indel population remains the same. We are not producing a gain of function mutation, so we're not creating a problem by disabling NRF2. This work continues on. It's the way we look at things specifically. Is there a phenotypic response in which a new protein appears or a new form of NRF2? And we look at these with all kinds of antibodies. And are we creating new types of modified genes, genetic mutations that suddenly appear because we've targeted? And the answer so far is no. And in addition, we don't see any reversion back to wild type, which is a fair question that we sometimes get. What we target and we knock on NRF2, specifically in tumor cells, that knockout pattern in the population of tumor cells remains the same. It doesn't go back to wild type. That's a big step forward for some of our FDA applications. So I just want to summarize a little bit about my thinking about how we chose the target and how we went forward. Careful choice of target, focus on an unmet medical need. Keep your endpoints modest and achievable. You really want to show proof of principle, especially in the area of gene editing, because the gene editing area is still innovative. We don't truly know if it'll ever work. We hope it will. We have all kinds of iterations of this, and lots of money are being put forward to commercialize it. But it would be very interesting to see if we can move forward on this. The way to do it is to build a strong scientific and believable foundation, and that's only achieved, as it always has been in science, by modest and achievable endpoints. Think about delivery vehicles, how best to deliver things. The tumor genome-specific targeting is our ace in the hole. We can distinguish between tumor and normal cells. And supportive biodistribution studies, low off-target mutagenesis events are very important, also to maintain. I just want to mention a couple things. We are committed to introducing medical technologies to minority communities, and we also promote women in science. The team below in that lower picture are the women that work at GEI, and there's a vast majority of them over men. We continue to push forward with that, and we'd like very much to continue to see more women enter science. And maybe the gene editing CRISPR field is the one that penetrates the overall population. These are our partners, whether they've been involved in producing or supporting the work, or in auxiliary program and in our education side. Here are the team leaders that have done all the work I've talked about today. Byung-Chun Yu, and there's Kelly Banis in the middle. Very, very young, but brilliant young scientist that leads our lung cancer program. And finally, a very important group of people. Early on, we developed a strong clinical development team with Amy Jennings, Holly Wang, Dr. Nicholas Petrelli, who chairs the scientific advisory board, and the guy that's probably going to drive this forward as the PI, surgical oncologist Greg Teasy. I want to thank you all for listening to this today, and hopefully we'll engage some very interesting questions. Here is all of GEI. Thank you.
That's great. Thank you very much, Eric. Another amazing talk, and I'm sure we've got lots of questions for you. I can see that we've had a few in already, so I'll move straight on to those. Okay, so your first question is, in your talk, you discussed the use of a non-viral LNP as a delivery vehicle for the CRISPR-Cas9 gene editing payload. Could you explain why you chose to take a non-viral delivery path instead of the more traditional viral delivery vector to introduce CRISPR-Cas9 into the cells?
Yeah, so that's a fundamental question that probably deserves another Tech Networks talk, Carolyn. I'm sure that many of us have had this, but briefly, it's both scientific and practical. Uh, Viral particles tend to be required to be made by CROs or CDMOs, and the expense is almost prohibitive, especially for a small research group. In addition, the quality of the viral particles, especially adenovirus or retrovirus, tends to be a bit challenging, and you never quite know what you're going to get. And then the cost is incredibly expensive. So to treat, just to give you an example, we did find a viral production group that would provide these viral particles for us containing our CRISPR-Cas payload, $4 million in 18 months to treat 10 patients. It's not sustainable. So we chose the non-viral particle, one, because we're getting great results with it, but two, it's really more like a drug, and people are very comfortable in producing that. So the turnaround time for that is approximately 90 days, maybe 3 months, 4 months. At a cost, it's about 1/4 the cost of the viral particle. If it were not producing the same efficacy and even better than our initial viral studies, we wouldn't have switched. But I have to admit that some of it has to do with the practical considerations.
That's great. Thank you very much, Eric. Moving on to your next question. Traditionally, gene therapy approaches to oncology was centered on targeting tumor suppressor genes or oncogenes. Have you ever considered following along this path instead of choosing NRF2 as the target gene for editing?
Yeah, this question kind of comes out of something I touched upon, and the answer is we did, and a lot of other people did too. Um, Tumor suppressor genes are quite complex. The shunt pathways and backup pathways for tumorigenesis are very complex as well. And so, if you try to repair a mutation in a tumor suppressor gene, first you'd have to get almost all of the genes, all of the tumor suppressor genes in the tumor repaired, and that is completely unachievable right now. Even with the knockout, which is the fundamental activity of the CRISPR-Cas molecule, 90 to 95% on some genes is definitely achievable. And for us, we're very lucky because that is true for NRF2. But other genes mysteriously are very resistant to that. So even the fundamental basis of what CRISPR does naturally doesn't give you 100%. Now you try to precisely repair or reverse the phenotype or genotype of a tumor suppressor gene, and it's simply unachievable. I hope it works. I mean, we're not going to take that route because we think it's a rabbit hole. But I do believe that if you target genes that control response to regular therapy, you'll be in a much better position.
That's great. Thank you, Eric. I think we've got time for one more question. And it is clearly a major discovery in your work has been the use of the PAM target sequence for CRISPR activity, where you have shown that it is possible to disable a gene in a tumor cell while not disturbing the same gene in a healthy cell. Since I believe you showed a slide that lists other possible targets, what are the scientific and clinical criteria for selecting the next target along those that have suggested are amenable for those sorry about this, amenable for such an approach?
Yeah, no, thanks, Carolyn. I understand the question. Essentially, when you get to a point where you've achieved good proof of principle from a bench scientist or fundamental level, and you're comfortable with mechanism, and that's always our first level. And so when we look at those other targets, we sort of start to think about ones that we can achieve. I don't think there's a lot of worry in our lab here that we can't target all of those genes pretty effectively in tissue culture cells. But now we have to think about, if we're going to apply that, delivery molecule, introduction of how it goes forward, and production level. So the clinical side is number one. On a practical side of that, followed up by how can you really help? Will, in fact, knocking NRF2 or KRAS, which is a gene that has the same kind of profile, EGFR, much more famous genes with this kind of unique mutation, if we were to knock that out, is there supplemental or augmentative therapy that would actually help in that case? So this is where being embedded in a cancer center with talking to oncologists who treat patients on a regular basis and are part of clinical trials on their own using Keytruda, Opdivo, and other types of targeted therapy and immunotherapy is really helpful because it helps us design the endpoint. So if one of those targets has an achievable endpoint and a fairly straightforward road, that would be how we would go forward on it. Achieving knockout of particular genes, we teach that in our educational program. We have high school kids who have never picked up a pipette before that use our educational kit, CRISPR in a box, that can do that. But it's the next step. How can you apply that, and will that have a true impact on the disease in a patient?
That's great. Thank you so much, Eric. And unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be made available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Eric, and we will be back soon with the next talk.
Very good. Thank you, everyone. Thank you, Carolyn.